This lecture is a review of the comparison between skeletal muscle and cardiac muscle. The major topics we're going to cover, we'll start with action potentials, then we'll get into excitation contraction coupling, or EC coupling. This is the connection between the action potential and the contraction machinery itself. We'll next talk about the fiber structure and how they relate to function. We'll get into motor units and how they relate to contraction strength, and finish up with the nervous control of both these muscles. Let's remind ourselves what an action potential is. It's just a way to get an electrical signal from one part of the cell to another. To do this, we're going to move ions across the plasma membrane. We'll first review how ions are going to cross plasma membranes. And we need to talk about two important types of membrane protein. If we recall, our membranes are made out of these lipid bilayers. These bilayers effectively stop ions from getting across, so we have to provide ways. The first way we're going to do this is using what's called a pump. Pumps are exactly what they say they are. They pump ions from one side of the membrane to another. To do this, they require ATP. These guys are always going to be on, and this is the only type we really need to worry about. This is the sodium-potassium pump, and it's doing two things at once. First, it takes sodium from inside of the cell and throws it outside of the cell. Potassium goes the opposite way. Potassium is pulled in from the outside of the cell into the cytoplasm. So this guy is going to set up our ion imbalance, sodium outside versus potassium inside. This is always going to be working in both of our types of cells, skeletal muscle and cardiac muscle. Let's talk about the next type of membrane, protein. These guys are going to be channels. Once again, they're just what they say they are. They're going to provide a way for ions to get across a membrane. We're going to see three different types. The first type is called a leak channel. And once again, it's just what it says it is. This guy is always going to be a little bit open. This one here shows a sodium leak channel, letting sodium cross the membrane. This is all going to be driven by diffusion. The diffusion is set up by the sodium-potassium pump. We won't see a sodium leak channel, but we will see a potassium leak channel just in a few minutes. Our second type of channel is a chemically gated channel. These guys are going to open and close when a neurotransmitter or a hormone binds to them. We'll only see this one time. For this discussion, it's not too important. Our third type of channel, and our most common, are voltage-gated channels. These respond, i.e. they open and close, in response to changing membrane potential, or the voltage around the membrane. At certain potentials, voltages, some will be open, at others they'll be closed, and we'll occasionally see ones that get inactivated. That means they can't open again for a while. But for this talk, I'm not going to mention these guys in any detail. All of these channels are specific to certain ions. This one happens to show a sodium one. But we'll see potassium ones, we'll see calcium channels, and we'll even see chloride channels, but not today. Next, we need to set up what's called a resting membrane potential. All this means is that we're going to set up a potential difference. We're going to make one side of the membrane a little bit positive and one side a little bit negative. To do this, there's going to be three things involved, and I'll do this in just a little bit of review detail. First, we'll start off right down here. We can mention again our sodium-potassium pumps. These guys are always working. There's no difference between the ones in skeletal and cardiac muscle, and they set up our ion imbalance. We'll have sodium ion outside of the cell and potassium ions inside of the cell. This alone doesn't set up our resting membrane potential because they're both positive ions. We're going to add two things to make our resting membrane potential. Next, inside the cell, there's going to be lots of negative molecules. We show a protein here, but there's many others. Third, we're going to add our one and only leak channel. And the only one that's important is this potassium leak channel. This leak channel is constantly open, but just a little bit. It allows potassium to slowly leak out of the cell. These three factors combine to make the outside of the cell a little bit positive and the inside of the cell a little bit negative. We can put an average number to this, and the voltmeter up here shows minus 70. That means the inside of the cell is a little bit more negative to the outside. This is a very small number, but it's enough for our purposes. we're going to see how we're going to change this number to send a signal from one side of the cell to another. We show a neuron here, but this can be applied to skeletal muscle. They're just about the same. Next, we'll go through a couple of important terms. We set up this resting membrane potential. We're going to call this a polarized state. One side of the membrane is different from the other. A depolarization event is simply a switching of our polarity. We'll make the outside negative and the inside positive. We're going to do this by opening and closing channels. Once we depolarize, we typically need to repolarize. And this is a return to our original situation, a reversal of our depolarized state. We can hyperpolarize, but for this lecture it's not too important, and we're not going to mention it anymore. There is a third state called hyperpolarization. This is when we will actually polarize to a greater extent. For skeletal muscle it does have importance, but for our comparison it's not that important. So a quick review of what we're trying to do here. We're sending an electrical signal from one part of the cell to another. We're going to do this by sending a wave of depolarization, and this is going to be called our action potential. All it's going to be is we send a wave of switching from negative to positive on the inside down from one end of the cell to another. And we're going to do this by using our voltage gated channels. This picture is a nice summary of how we're going to do our action potential. Just to remind ourselves, up here we show our sodium-potassium pump, setting up our ion imbalance. This guy will never stop working as long as there's ATP available. This will reset our ion imbalances after action potentials pass by. The first thing and the most important to start our action potential is our voltage-gated potassium channel. At resting membrane potentials, this channel is closed. But once we depolarize this channel above what's called a threshold number, this channel opens. And the opening is an extremely fast event. Diffusion will drive sodium from the outside to the inside of the cell. Remember, the resting membrane potential says the inside of the cell is negative. As sodium comes in, it will depolarize this inside and actually make it a little bit positive. To continue, this local depolarization will be enough to set our next set of sodium channels above our threshold value. These guys will open, sodium comes in, and will activate the next set. This is going to have the effect of having a wave of depolarization travel from one set of sodium voltage-gated channels to the next set and on down the membrane. Behind this wave of depolarization, we must have a reset. We must have a repolarization. Now we can look at our next set of voltage-gated potassium channels. Once again, at resting membrane potentials, these guys stay closed. But once we depolarize this, these guys will open. They're a little bit slower than the sodium-gated channels. These things are extremely fast opening and closing. Once we will have our depolarization phase and the sodium channels close, our voltage-gated potassium channels will open up. Potassium is driven the opposite way by diffusion, so potassium will go from the inside to the outside. This has the effect of resetting our inside depolarized state back from positive to negative. The overall effect here is our wave of depolarization, traveling from one end of the cell down to the other. Each set of voltage-gated sodium channels will set off the next set. Our local depolarization will set off the next set and on down. Behind our wave of depolarization is our potassium channel-driven wave of repolarization, resetting to our normal resting membrane potentials. You need this because you probably need to send another action potential very soon after this. Now let's look at a slightly bigger picture. Once again, we have our two sets of channels. We have our voltage-gated sodium channels and our voltage-gated potassium channels. Sodium channels are for depolarization, potassium channels are for repolarization. This shows a plasma membrane of a skeletal muscle. This is also called the sarcolemma. This is going to send a wave of depolarization, our action potential, from one part of the cell to another. We're now going to start to consider how we have our excitation-contraction coupling. We need to find a way to connect our depolarization, our action potential, with our actual muscle contraction machinery buried deep within the cell. First, we're going to need to get our action potential from the surface of the cell all the way to the inside of the cell. We can do this in muscle cells by what's called a T-tubule. This is just a big tube running from the surface of the cell down into the middle of the cell. Our action potential will have no trouble continuing down this tube, and that's going to get our action potential from the sarcolemma all the way deep within the cell. The next step is going to be another set of tubes inside of our cell. Right here we see what's called our sarcoplasmic reticulum. If you remember back to 141, this is actually the same thing as smooth endoplasmic reticulum. This is acting as a calcium storage. Jumping ahead, calcium ions are the last signal. These guys are going to tell the muscle machinery to contract. Our action potentials will dive down our T-tubule. This wave of depolarization will actually jump across from the T-tubule membrane to our sarcoplasmic reticulum. The sarcoplasmic reticulum acts as a store of calcium. On the surface of the sarcoplasmic reticulum are another set of voltage-gated channels. These are calcium channels. The depolarization will open these. Once again, diffusion will drive calcium from the inside of these tubes out into the cytoplasm. Calcium will then hook up with the contraction machinery itself and complete our excitation contraction coupling. Next, let's quickly review the actual contraction machinery itself. We're going to be talking about our filament structure and the sliding filament mechanism. The two filaments we're going to talk about are our thicker filaments and our thinner filaments, and we can do this in light detail. The thicker filaments are made of this protein myosin, and these guys have two parts. They have a myosin tail and the actual contraction machinery itself, the myosin heads. If they can, the myosin heads will grab onto the thin filament. The thin filaments are mostly made of actin. Actin here is acting just like a rope. If allowed, the myosin heads will grab onto the actin and pull themselves along, and that's going to have the effect of shortening the muscle units. The name of the muscle unit up here is a sarcomere. This is one section. This type of muscle, both skeletal and cardiac, are very organized, and you can see the banding pattern caused by our sarcomere structure. The last bit of detail here is down on our thin filament. The thin filament, again, is mostly made out of actin, which is just acting as a rope. But we have these two other proteins here which control whether myosin can grab onto the actin, and they are our troponin and our tropomyosin. With no calcium around, the tropomyosin blocks access from the myosin heads to grab onto the actin. But in the presence of calcium, once it's released from that sarcoplasmic reticulum, calcium will bind to troponin, which changes shape, shifting tropomyosin away from the actin. So if calcium's around, troponin shifts the shape of tropomyosin, the myosin heads can grab onto the actin and they will pull themselves along the actin, shortening the muscle. The effect is something like this. With no calcium, you will see the sarcomere in its relaxed state. We release calcium from the sarcoplasmic reticulum. We make the actin available. The myosin can grab onto the actin and it'll pull itself along, shortening the sarcomere and contracting the muscle. So a quick review of the whole process. We start over here with our action potential. A depolarization above threshold will open our voltage-gated sodium channels, which cause the depolarization. Local depolarizations will depolarize the next set of sodium channels. We will send a wave of depolarization along our sarcolemma, or plasma membrane. Behind that is our wave of repolarization. The potassium channels, again voltage-gated, will open up just a little bit behind the very, very fast opening and closing sodium channels. We will send our action potential down the T-tubule, which crosses over into the sarcoplasmic reticulum. Our voltage-gated calcium channels will open up, releasing calcium into the cytoplasm. If calcium is present, we will shift our troponin, tropomyosin complexes, allowing myosin to grab onto the actin, and they will pull themselves along, shortening our sarcomere. If we stop sending action potentials, the calcium will be reclaimed by the sarcoplasmic reticulum. Once calcium is reclaimed by the sarcoplasmic reticulum, we will block access from the myosin heads to the actin and the muscle will relax. We can sum up the electrical activity of the action potential using our voltmeter in a quick chart. Just to orient ourselves, over here we will have the time. Notice the time scale. This is very important. This time scale is in milliseconds. This is 1,000th of a second. On the left, we will see our voltmeter, our millivolt scale. We'll start off down here at resting membrane potential, approximately minus 70 millivolts. We'll first depolarize a little bit. Once we get above threshold, this sharp spike here is our depolarization spike. This is caused by sodium channels opening. Sodium channel rushing in, causing our depolarization. At the top here, our sodium channels will very quickly close. About the same time, our potassium channels will opening, repolarizing back to resting membrane potential. Potassium channels stay open a little bit too long, so they'll let a little too much potassium out of the cell. This will have the effect of this hyperpolarization event. The last word to mention here is there is a little time period around here where you cannot reactivate this cell, and this is called our absolute refractory period. We can combine our action potential with our actual muscle contraction graph. Once again, let's orient ourselves with a time scale. From here to here is just a few milliseconds. The blue line is showing our action potential, our very sharp depolarization and our just as sharp repolarization. This shows us the muscle contraction for a skeletal muscle, and this is called our muscle twitch. Just a brief contraction and then a muscle relaxation. All of this is going to be under about 100 to 200 milliseconds, a very fast event. Remember, skeletal muscle, you must be able to contract it extremely fast. But just as importantly, you also need to relax it extremely fast. So the action potential for skeletal muscle will be extremely brief. If we just want to keep this muscle contracted, we can keep sending action potentials, and this muscle will stay contracted. Now we build a picture of how skeletal muscle functions. Let's compare this to the cardiac muscle function. Some things will be very similar or identical. Some things, though, will have some significant differences. Let's first look at our cardiac muscle cell sarcolemma, the plasma membrane, and see if the channels are the same or different. The first one we can see here is our voltage-gated sodium channel. This is essentially identical to the one in skeletal muscle. We're going to add a word here in the front, though. This is a fast voltage-gated sodium channel. Remember how fast this guy opens and closes? So this guy is going to be called a fast version. We will see a slow version of this sodium channel. This is responsible, again, for our very fast depolarization phase of the action potential, and it's identical to skeletal muscle. The next, though, was our potassium channel. This is not the same as in skeletal muscle. We've added a word in the front here. It's a slow voltage-gated potassium channel. In skeletal muscle, our voltage-gated potassium channel opened just after the voltage-gated sodium channel closed. This guy is even slower. It actually doesn't open for a couple of 100 milliseconds after our voltage-gated sodium channel closes. We're actually going to add a third channel here. This is going to be a slow voltage-gated calcium channel. This is going to affect our action potential in a very significant way. This graph is for our cardiac action potential, and you can see it is going to be different from our skeletal muscle action potential. The first thing, however, is the same. Our depolarization phase, caused by our very fast sodium voltage-gated channels, is exactly the same. Once we depolarize above our threshold value, this channel will open up. Sodium floods in, a very fast depolarization. Very, very quickly, maybe a millisecond after opening, our channels will close up. This is going to stop the increase in depolarization. Next, we see our first significant difference. If you remember back to the skeletal muscle action potential, the next event was our voltage-gated potassium channel opening up just behind our sodium channel, and our very quick repolarization event. Remember, this spike went very, very quickly down and even hyperpolarized a little bit. We do not have the same voltage-gated potassium channel here. This voltage-gated potassium channel doesn't even bother opening up for almost 200 milliseconds. If you see the time scale going from 0 to about over here, this is when we will have our repolarization phase. This is when our very slow voltage-gated potassium channel opens up and repolarizes. We really don't have any hyperpolarization here, as this guy is so far delayed behind the depolarization event. It may not sound like much, but this 200 milliseconds, about 0.2 of a second, is a very significant time period. Next, we're going to add our new channel, our voltage-gated calcium channel. Once we depolarize, we would pretty quickly repolarize. We would do this because of those sodium-potassium pumps would reset our membrane potential. It turns out we actually want to stay in our depolarized state, and this is what our calcium channels are going to do. These guys are just another set of voltage-gated channels, but they are channels for calcium. They will open up right around here, just after our depolarization event. Calcium will go ahead and enter the cell. This is the same thing as sodium enter the cell, so this will actually maintain our level of depolarization. This keeps this cell depolarized. This flat area here on the action potential gets the name our plateau phase, and this is definitely not seen in skeletal muscle. It is unique to cardiac muscle. This has the effect of prolonging our depolarization out to about this 0.2 seconds or 200 millisecond time period, and then we will repolarize back to resting membrane potential. The important question is why? Why do we need to maintain this depolarized state or plateau phase in a cardiac muscle cell? This slide shows you the entire picture of our depolarization, plateau, and our repolarization action potential, and then our muscle contraction and relaxation. There's a simple reason for this elongated action potential. What we don't want to allow is our cardiac muscle just to have a very brief contraction, kind of that muscle twitch. That will do us no good. Why? Because remember, we're trying to eject blood. We need to squeeze that blood, generate blood pressure to get sufficient ejection from those ventricles. To do this, we cannot have a very brief contraction. It's got to be a certain length. And once again, we see this length of about 200 to 200 milliseconds, about a quarter of a second. This depolarization, followed by this longer muscle contraction and relaxation than skeletal muscle, will make sure we get enough contraction of cardiac muscle to have sufficient blood ejection. The other part of this is our absolute refractory period for cardiac muscle is much longer. This will prevent extra stimulation coming in and sort of confusing this muscle. From here to here, this muscle cannot be restimulated. This sort of sets a speed limit on the stimulation of the cardiac muscle. Remember, we must have sufficient period for this guy to contract, to eject blood, but also we need time for this guy to relax and have sufficient time to fill with blood before the next contraction. How does the excitation-contraction coupling events compare between skeletal and cardiac muscle? Just to make it easy, these two events are relatively similar. Once again, we do have T-tubules in cardiac muscle. We also have sarcoplasmic reticulum, which functions exactly the same way as skeletal muscle. It responds to action potentials and releases calcium into the cytoplasm. The easy part of this is the contraction machinery is almost identical. Remember, both these type of muscles are striated. They both have very similar sarcomere structures. They both have thin filaments and thick filaments. And the contraction machinery and the control is close enough that for this class we can just say it's identical. The only minor difference is that there's less T-tubules in cardiac muscle. Do remember that we have added a calcium channel to the sarcolemma for cardiac muscle. So calcium actually comes from two places now. In skeletal muscle, it is only going to come from the sarcoplasmic reticulum. In cardiac muscle, calcium also enters through the calcium voltage-gated channels from the outside of the cell to the inside. For this reason, we just have less sarcoplasmic reticulum. It's not as important as skeletal muscle. That completes this section. Next, we'll talk about how the structure of the muscle affects its function.